Okay, so line of data there is 7B71, line 22, 7B71, Helices, Spectroscopy, Popper Interface, 5G Wow Seti, there's the alien radio signal. This is from March 15, 2013, today's June the 24th. This is about interface controls, the UFO spaceship and the wild data. It's a long video and it's from one PDF file of results. So March 15th, this is from the Ligand PISEMA cell compounds found in 7B70. SAM14 is a pulse sequence to PISEMA. June 11, 2030, keywords from the Dennis PISEMA and SAMMY experiments. www.ncbi.nlm.nih.gov. Quotes from the blogs are from December 2011, from 2006. Structure determination of a membrane protein with two transmembrane helices in aligned phospholipid bicelles by solid-state NMR spectroscopy. More scientists in www.ncbi.nlm.nih.gov. Abstract. Structure of the membrane protein MRF-FT was determined in magnetically aligned phospholipid bicelles by solid-state NMR spectroscopy, with two transmembrane helices and a 10 residue interhelical loop. This truncated construct of the mercury transport membrane protein MRF has sufficient structural complexity to demonstrate the feasibility of determining the structures of polytopic membrane proteins in their native phospholipid bilayer environment under physiological conditions. PISEMA, SAMMY, and other double resonance experiments were applied to uniformly and selectively 15N labeled samples to resolve and assign the backbone amide resonances and to measure the associated 15N chemical shift and 1H to 15N heteronuclear dipolar coupling frequencies as orientation constraints for structure calculations. 1H13C 15N triple resonance experiments were applied to selectively 13C and 15N labeled samples to complete the resonance assignments. Especially for residues in the non-helical regions of the protein, a single resonance is observed for each labeled site in 1 and 2 dimensional spectra. Therefore, each residue has a unique conformation and all protein molecules in the samples have the same 3-dimensional structure and are oriented identically in planar phospholipid bilayers. Combined with the absence of significant intensity near the isotropic resonance frequency, this demonstrates that the entire protein, including the loop and terminal regions, has a well-defined stable structure in philosophic bilayers. So the keywords are MRF, bacterial mercury detoxification system, bilayers, PISEMA, SAMMY, PISA wheels, dipolar waves. www.ncbi.nlm.nih.gov. Early experiments demonstrate that solid-state MR spectroscopy is capable of determining molecular structures with atomic resolution. The following Popper user interface control may not be accessible. Tab to the next button to revert the control to the accessible version. Destroy user interface control 9. Protein structure determination by solid-state MNR of aligned samples is based on the narrowing of resonances that can be achieved for immobile molecules of multiple pulse. The following Popper user interface control may not be accessible. Tab to the next button to revert the control to accessible version. Control 10 and double resonance. The following Popper, it just keeps saying the same thing over and over. Experiments, the spectroscopic simplifications associated with sample alignment, the following, I just keep saying it's not accessible. That's weird. Okay, so destroy is transported across the inner cell membrane into the cytoplasm by membrane proteins such as MRF. And it's got just the amino acid resequence of the truncated construct MRF used in the experimental studies is compared to that of full-length MRF. And then here's the diagrams for it. 2-dimensional 15N chemical shift, 1H 15N dipolar coupling NMR, spectrum of uniformly 15N. Composite spectrum consisting of PISEMA spectrum, SAMMY spectrum, chemical shift axis B. There. Representation of the 2-dimensional spectrum with the centers of the circles corresponding to the values in Table 1, which were obtained from the best available measurement. The diameter of the circles corresponds to the estimated experimental uncertainty in the measurements. The resonance assignments are noted. Colors are noted to denote resonances with segments of the protein www.ncbi.nlm.nih.gov. Plot of experimental dipolar coupling frequencies as a function of residue number with dipolar wave fits. The frequencies and are divided into 4 groups that are color-coded by residue number in the primary sequence of the peptide corresponding to the secondary structure domains of the protein. N-term resides, loop resides, second helix resides, blue wave, and C-terminus purple. There's the diagrams there. Flow chart for the structure calculations, Ramakide plots, Rama of prolines, prolines, lysines, and all other residues used to restrict the space of the torus and angles for non-helical residues. Definitions of the parameters for the helix-helix packing constraints described in the text, images in this article. Three-dimensional backbone structure of residues, 27 to 68 of Murph feet in philosophical bilayers, wireframe representation, ribbon diagram, back-calculated spectrum dots from one of the structures shown in Part 8, superimposed on the representation of the experimental data, Ramachandran plot with all the backbone angles marked with dots. Keyword SAMP14 is a pulse sequence. Will be in the next video. Thank you for watching.